So I'm not going to give you. I was saying, Carlo just asked me to make a comment or two, so I haven't prepared a lot of detailed discussion, but maybe it would be interesting for people for me to comment almost like historically. I think where I want to start is actually at the Pasteur Institute. You know, our field depends initially, I think, on, at least in large part, on the way people studied teratomas. And Francois Jacob, who's extremely famous for his work in molecular genetics, his group at the Pasteur switched when they figured out that they understood how genes worked to look at how teratomas work. And they were very interested in the idea that teratomas make all the cells in the body. And that really captured my attention. And at the time, really working with cell lines wasn't a popular thing to do. And not just was it not popular, it was regarded as being sort of slightly lunatic. Cell lines were regarded as being artifacts, basically. People felt sorry for your mother if you studied cell lines because her son had deviated from any sane path in their professional life. So the idea of making dopamine neurons stems in part from the sort of fundamental discovery actually made in Sweden that you could recognize dopamine in tissue very readily because if you treat tissue with formaldehyde, dopamine becomes autofluorescent. So you could see where dopamine was. And so the idea of making dopamine neurons in a dish was prompted partly, of course, by the use of fetal tissue in Parkinsonian trials. But there's a really complicated history there, which I won't go into. But pretty quickly, we realized that you could actually make, from the mouse, embryonic stem cell, which is close enough to teratoma for this purpose, you could make dopamine neurons. And that prompts me to make two comments, really. So one is, it's of course extremely important issue, making dopamine neurons, for all kinds of reasons, one of which is transplantation. But if you look at, for example, at the gene which we're going to discuss later on in the presentation today, the dopamine transporter SLC6A3, and you start thinking about what does this protein do, the answer is it does all kinds of different things depending on exactly what context you're looking in. By all kinds of different things, I mean all kinds of clinical things in humans. And so we became very interested in the dopamine system very early on. And in our group, we first made it for mouse ES cells, and Lorenz was in our group at the time, Lorenz Studer, who's gone on to be a real pioneer in this field of applying this in vitro stem cell derived system to Parkinsonian models and now increasingly in Parkinsonian patients. So I think I'll just close by making one other comment, which I think is relevant to our discussion today, which is going to center around, as you can see in the title here, the spatial and temporal differentiation of midbrain neurons. That if you look in the mouse, at least, or the rat, dopamine itself is made in large amounts as soon as the neurons differentiate. And dopamine neurons in the midbrain are some of the first neurons to differentiate in the forebrain. And actually, some of you on the call may pick up on this because we've sort of talked about this before, that the role of the dopamine, the developmental role of the dopaminergic system is extremely interesting to think about. And I remember when I was once visiting the Karolinska Institute, where they did a lot of this chemistry. And I asked Lars Olsen, who I was speaking to in his office, when do you first see dopamine itself in the brain of a rat, I guess he was looking at. And he took a picture from his desk of the Hill-up reaction, the reaction that was used to detect dopamine, which was of the very earliest stages of midbrain differentiation, which was shining with dopamine. And he said to me, "This is from my PhD." So I just want to say to you all that the topic we're discussing today is very interesting for a whole number of reasons. I'm very much looking forward to listening. So well, I hope that's useful, Carla.
Thanks, Ron. So now from the historical perspective, we can jump into what's going on right now. So Pau has done a lot of work in different areas that are of interest to this group. And today he's going to be focusing on his work in the midbrain dopaminergic system, both in vivo and in vitro. So thanks, Pau, and really excited to hear what you're going to share with us.
Yeah, the first thing I want to say is that please, Ron, if I say something that's not totally correct or wrong, jump in and tell me, because I think you are the expert here, not me. Okay, so before starting this talk, I would like to thank Carlo for organizing this symposium. I think this is a really nice community around development, and I hope this will foster collaborations in the future. So thank you for doing that. And as said, today I'm going to talk about the midbrain, not the forebrain, not the cortex. It's a bit different as the usual discussions here. Another thing I wanted to highlight is that initially this project was envisioned as an atlas, but I think the reviewers didn't like the idea that we just had a few lines and a few fetal samples, so we had to reframe it a bit. And rather than presenting this resource as based on the width of the data set, we profile it to the depth. So we try to use different tools to try to understand as many things as possible from the first and second trimester of midbrain development. I would like to also highlight the main contributors of this project. This is a kind of project that has bridged my postdoc in two different places, first in London and later on in Helsinki. So it was done at UCL and Helsinki Institute of Life Sciences. And it would have not been possible without the other two co-authors, Dimitri and George, which have done one the experimental part and the other spatial part. And of course, the supervision and advice from the PIs, Sergey and Helena, which have been the two PIs of my postdoc, and Manju and Serena, which are part of the three group that has contributed to this research. So I could probably skip this slide since this audience know much better than myself what's human brain development. But just as a quick recap for the midbrain, you know that during the first trimester of brain development, and specifically around the 3 post-conceptional weeks, the neural tube closes and becomes segmented in 3 different compartments along the rostral caudal axis, the neural tube, the forebrain, the midbrain, and the hindbrain. The midbrain is the star of our story today. And is it indicated in green here? And if, for instance, we let it regionalize, pattern itself, it starts forming the different nerves, the different tracks, the different nuclei, which are involved in vision, sensory processing, cognitive function, motor control, a lot of things. But the particular, and actually I would say the emblematic view of the midbrain is this coronal section that I'm representing here, where you can see the different nuclei, the cerebral aqueduct, the red nucleus, the substantia nigra. And probably the reason why we started this project was to see how and to analyze how this migration of neurons and the maturation of neurons occurred. First with a kind of radial migration from dorsal to ventral positions, and later on throughout the substantia nigra in a more tangential migration. So that was the main reason to study which were the gene expression dynamics, what was going on in the midbrain during those times. As this community knows very well, studying the brain is quite difficult because it's a very inaccessible cell type, especially considering in development. That's the reason why many of us are using in vitro models, either 2D or 3D. But using those tools also come with its own strengths and limitations. For instance, 2D is very good at generating homogeneous monolayers with high cellular purity, but they somehow lack the spatial context. While 3D organoids can provide that, and they can also be cultured for longer, they have the problem sometimes of reproducibility, and they tend to present a necrotic core because there is no good enough diffusion of nutrients and oxygen in the inner part. So what we wanted to do, first of all, is to benchmark these 2D and 3D models versus fetal samples to see what kind of features could we actually recapitulate of in vivo development. And in addition of that, we wanted to model a disease, the DTDS syndrome, which is the dopamine transporter deficiency syndrome that, as Ron was already mentioning before, is caused by compound heterozygosity in the dopamine transporter gene. And our idea here was to generate 3D organoid models to analyze which kind of features could we learn from the developmental midbrain in that particular case. So the aims of this study were three: mostly understand the diversification, migration, and maturation of dopaminergic neurons, to compare the 2D and 3D versus the fetal, and also to perform some kind of disease modeling with the 3D organoids of this Parkinsonian disease, the DTDS. For that, we have used a multimodal approach, combining different kind of techniques. First of all, we have used histological imaging to characterize the anatomical changes during fetal development, and also during in vitro differentiation. We have also generated a single cell dataset to define the different cell type identities and to systematically evaluate the models, including control and patient lines. And we have also used spatial transcriptomics to capture the gene expression dynamics in their spatial context, with a particular focus on the migratory programs in the developing midbrain. So now I will look through in a chronological order for each of these tools that we have used. The histological analysis doesn't have much history. It's essentially tracking the key protein markers using immunofluorescence and during midbrain development. And that's what essentially the work that Serena, Dimitri, and Charlotte have been doing. So to characterize the in vivo spatiotemporal emergence of this midbrain complexity, we have provided fetal samples from 6 to 22 post-conceptional weeks, which more or less corresponds to the time that the neural tube starts to regionalize itself to where we can start seeing patterns of neuronal maturation. What we saw is that at 6 post-conceptional weeks, the fetal tissue already expresses key transition factors, SOX2 and LMX1A, which indicates that there is the presence of a population of proliferating neural progenitors in this ventricular zone. During the second trimester, this ventricular zone progressively expands and starts to organize itself. It's highlighted with this much more abundant green all around. And by around 19 post-conceptional weeks, the red nucleus become a bit more apparent. And red nucleus is lateral to the ventral midline, which is the path that neurons follow for the first kind of radial migration. Later on, at 22 post-conceptional weeks, we see that the midbrain complexity increases even more, and we have now very well observed, thanks to the TH neurons, the structure of the substantia nigra as well, the ventral tegmental area. But what was really relevant here was to see how this neuronal migration and maturation was happening. And that's mostly determined by the position of dopaminergic neurons, which are marked in red by tyrosine hydroxylase here. Initially, GFAP positive cells, which are marked in green, are located along this ventral midline, which following this radial distribution that's indicated by number 1. And they keep this kind of elongated morphology for 13 and 19 post-conceptional weeks. But at 22 post-conceptional weeks, they somehow lose this kind of directionality. As for TH positive neurons, we see that they are present, but it's not until 22 post-conceptional weeks in which they start to adopt this kind of morphology, but now not along the radial, but the tangential migration. So what is also interesting is that both GFAP positive and TH positive cells colocalize in that sense. So this is suggesting that for this tangential migration, we also need GFAP positive cells to be present for the neurons to mature. In addition of that, we have used other markers like noon, which highlights that a fraction of the TH positive neurons are actually also expressing this super mature marker. So a fraction of the TH positive neurons are more mature than others, indicating that during this migration, during this process, there is different levels of maturity along the substantia nigra, at least at this time point. But all we have seen until now are histological images of the in vivo data. What does it happen in vitro? To try to answer that, to try to see whether we can recapitulate the main events or the main cell types that we have seen, we used control lines that were differentiated in either in 2D or 3D between days 20 and 70, and for 3D we could get another sample at day 120. For 2D models, we could confirm the efficient neuralization and ventral midbrain identity. We again observed the presence of the progenitors, FOXA2 and LMS1, as well corin, which is somehow a marker for ventral floor plate dopaminergic progenitors. The presence was both in 2Ds and 3Ds. Here, MLO indicates midbrain-like organoids. The difference was starting to appear at day 40 when we look at post-mitotic neurons. In this case, in 2D, we can observe that there is a clear abundance of MAP2 positive neurons, while in 3D, there is a mix of MAP2 and TH positive. There is much more red in this image than in this other image. So somehow the amount of TH neurons is higher at day 40 in 3D organoids. And when we look at the extreme of that, we can detect the presence of many different neuronal markers, ADC, GABA, CYP, in the midbrain organoids at the day 120. And actually, if we look at the entire organoid, we can see that there is 9 and TH neurons, which project the neurites to the outer part. They have the neural crest, and after performing a Fontana Mason staining, we can detect the presence of melanin, seen as this dark pigmentation in the image. So that's somehow ensuring that we can achieve a mature profile of neurons by this time, which somehow confirms that with 3D organoids, you can recapitulate some of the in vivo biology, at least to some degree.
Can I ask you a question about that image,
yes.
just to keep the discussion going? So you commented on the red nucleus. So do you find, are there red nucleus neurons present in this system at this time?
That's a very good question, and I'm not sure we specifically look at the red nucleus. I can ask the other collaborator, but I don't think we specifically try to address this question. But that's a very good point, because it might have a role too in some
yeah, sure.
kind of yeah.
Great, thank you.
Yeah. We will then try to do a kind of more high throughput analysis using single cell transcriptomics, and in this part, what we essentially profiled was control lines in 2D and 3D, again, same day 40 and 70, and an additional day 124 3D. And for the fetuses, we profile 4 samples, 2 corresponding to the first trimester and 2 corresponding to the second trimester. The second kind of samples we profiled were the patient-derived lines from these DTVS patients, jointly with 2 age-matched controls. And in addition of that, we added one of the patient lines was corrected, so we had one isogenic control. So we could somehow assess the temporal progression of those different models in 3D. And finally, to confirm or to actually observe the commonalities and the differences we have between our dataset and all the available published midbrain datasets, we perform an integration and look at how things work together. So initially, we focused on dissecting the cell type compositions of control cells only. On the left image, you have the UMAP embeddings of the cells annotated with a 2-tiered system. So in the big font is essentially the kind of low clustering resolution, which we have 10 different broad categories. and the small subtypes are the kind of high clustering annotation. The upper left, so we profiled around 35,000 cells from 6 donors. 2 of them correspond to the lines and 4 to the fetal samples. The cell types that we identified were essentially distributed in different groups. Here we have the big cluster of neurons, immature and mature neurons, which are shown by blue and green. And then we have another cluster of early and late midbrain progenitors, with a fraction of these late midbrain progenitors also committing to astrocytes. And then, as expected, we also have microglia. We have also perivascular cells, as well as some highly proliferative neural stem cells. Probably these kind of perivascular cells they come mostly from the fetus. When we look at the cell type proportions for the different samples and at the different time points, we observe that in vitro models produce much more neurons than the ones that are present in fetal samples. That's known, that's like how in vitro models perform. And instead, fetal samples have much more microglia and late midbrain progenitors than the in vitro models. To formally account for that, to formally account for this differential abundance of cell types, we use MILO-R. MILO-R is better than other differential abundance tests in the context of differentiations because it allows to model these continuous processes. And the first thing we did was to compare 3D versus 2D, accounting for the differentiation time point as a covariate of the test. What we observed was that there were one particular subtype of the dopaminergic neuron, as well different precursors that were more abundant in 3D versus 2D. There were also some cell types like these progenitors, which were enriched in both directions, which means that there is substantial cell state differences between the 3D and the 2D. And what to me was more interesting from all of this is that when we look at all the subtypes of dopaminergic populations and we perform differential expression between the 3D and the 2D, all of them converge with the similar Go terms. And that essentially is that there is an upregulation of the mitochondrial respirosome in 3D. So all the neurons seem to have a much more prominent role of the mitochondrial metabolism in 3D. We then perform differential abundance between the fetal and the 3D. In that case, we observe even more differences than when we compare the 2 in vitro models. That's somehow expected. There is much more differences between fetal and 3D than 3D versus 2D. And in this case, there were many changes. Microglia was more abundant in fetus. There were different populations of progenitors, radioglia that were more abundant. But there were some subtypes of dopaminergic neurons that were more abundant in 3D. And I was especially interested in this subtype too, to see what was the kind of the differences between the fetal and the 3D. And for that, we ran a differential expression and found that actually there is an upregulation of synaptic-related pathways or terms in the fetal cells when we compare them with the 3D one. So that somehow indicates that despite having less cells of this particular subtype, less dopaminergic neurons of this particular subtype, they seem to have a much more mature role, and at least in terms of synapses. So the quantity not always matters. It also matters the specialization and the maturity of it. And finally, in this kind of differential abundance testing, we also look for differences between the second trimester and the first trimester of the fetal samples. The results here were not really surprising. We observed an enrichment of microglia in second trimester and a kind of enrichment of neuroplasts of immature neurons in the first trimester. So not really surprising what we found here.
And can I ask another question? Just
Yes.
to keep us all thinking together. So
Yes,
if we
yes,
just
please
go
interrupt me
back
whenever
a couple of slides.
Yep.
That one, yeah. I mean, this is very, very interesting. So, I mean, I have two questions, I think. So one is, could you explain to us just in a sentence or two what these DA1A, 1B, and DA2 cells are? Are they related to the substandard classification of dopamine neurons in the midbrain, or is this a special new kind of?
Yeah, that's a very good question, and I forgot to explain it that well. So I really thank you, Ron, to ask me this. So whenever we had this challenge of annotating the cells, we said, okay, what's already out there, that's published, and that has tried to provide as much knowledge about it as possible. And we found the Lamano annotations at that point, which was one of the first midbrain efforts and the first single cell datasets. And we tried to use kind of the same markers that they used to annotate them. So this is the dopaminergic subtypes defined here are aligned with the Lamano ones, and they use the same markers. I cannot recall exactly which is the marker for each specific subtype, but I have a supplementary figure with the corresponding marker. Okay, so we can go back at this if you are.
No, that's helpful. Thank you.
Yeah.
And along the lines of this, so what you're sort of saying here, at least with, I think you said the DA2 class, but is it true for all of them that they're enriched for like synaptic gene expression, or just specifically for DA2?
At that moment when we were doing the study, we were specifically interested in this one because we thought that this one was the one that was associated to most maturity. But I'm pretty sure you are right. I am pretty sure that there is also more synaptic enrichment for the other two. I have not explicitly tested, but I am quite convinced this is the case too, because the expression profile of the subtype 2 and the subtypes 1A and 1B is very similar. The difference is very, very, very small.
I see. And this tyrosine hydroxylase enriched in these new cell types?
They express it, yeah, for sure. If it's
Right.
enriched, I'm not sure. We could test that. That's not difficult. Yeah, we could test that.
Okay, great. Thank you. Thanks very much. That's perfect.
Okay, so going back to until this point, I've talked about differential abundance. Now, what we start is trying to dissect the gene expression dynamics on the trajectories. The fact that we have fetal cells from the first and the second trimester provides us with a kind of unique opportunity to reconstruct the developmental trajectories using cell time analysis. And for that, we borrowed an approach that was used in a previous paper of the cortex from Niccolò Caporale from Tested Group, in which they use a graph approach. They essentially manages to isolate the different lineages, either the neuron one or the astrocyte lineage one. To do so, what they do is essentially to put the cells in a graph and then recluster the different cells at much higher resolution than the one you used for defining the annotation. And by doing so, you have these different nodes. You have to obviously set one anchoring node, one starting node, and then the end nodes. And then there is one algorithm that allows you to find the shorter path between your anchor and your final differentiation. So that's how we end up drawing differentiations toward from the early midbrain to the mature neurons, on from the early midbrain to astrocytes. I was particularly interested in the neural lineage, so all the results that I'm going to present today is about this lineage, but the paper also contains some of the astrocyte gene dynamics if you are interested. Not surprisingly at all, when we perform a principal component analysis on the cells that have been isolated for the neural lineage, what we capture is essentially the expected emergence of the cell type identities across the trajectory. That was what we were looking for, because if we can see that there is a significant correlation of the PC1 with the emergence of the cell types, we can then look at the gene loadings of this principal component and start thinking about if these genes make sense. And actually, we found that there were STATMIN and also synaptogene, so genes that are more linked to the neuronal state, which are actually important for the neurons. Moreover, if we look at the bigger picture and we look at all the genes that we have for this lineage, we can see that there is transition factors, LH1 and OTX2, which are also somehow more linked to the mature neuronal stage. On the other side, we have Wnt5a, which is, if I'm not mistaken, a non-canonical Wnt ligand, and SSH morphogenes, which are more somehow expressed in the early midbrain progenitors. Seeing this, we start to think that it was very important to characterize the gene expression dynamics, considering different types of genes, either enzymes, ion channels, morphogenes, transfer factors, transporter. To actually assess the significance of the change of the expression, we use a tool that's called TradeSeq, which performs differential expression, but on a trajectory. And in this case, I'm just showing the significance for the difference between the start and the end of the expression, but there is different ways in which we can apply this tool. The overall message from this analysis is that some of the somehow canonical, maybe Ron will have a different opinion on that, but somehow one of the canonical markers for dopaminergic neurons, TH and JIRC2, seem to peak at the middle of the trajectory rather than at the end, and that somehow surprised me. And other markers like OTX2, which to me were usually associated with more progenitor populations, seem to peak at the end of this trajectory. So that was quite interesting because it could reflect that there is a new role this gene in later differentiation, or at least at some point in differentiation, which I was not expecting. We then start to look at the patient line. So as said before, we have seen that there is some differences between the 3D and the in vivo composition. So we were wondering, are we able actually to study or understand something from the 3D? Can we capture any phenotype? And for this reason, we generated organoids from these patient lines together, as said before, with the control and the patient-corrected line. I'm not sure if I mentioned before, but very quickly, this syndrome is caused by compound heterozygosis in the dopamine transporter gene. And it usually, in this particular disease, what it happens is that there is an accumulation of dopamine to toxic levels, which ends up generating an apoptotic neurodegeneration in the system. So what we wanted to study is this kind of neuronal loss was happening. So we used ICC in this case, quantification of TH positive neurons. And as you can see at day 40, there is no difference. But when we look at day 70, the patient lines clearly have a reduction of TH positive neurons. So they have much less dopaminergic neurons. However, the patient-corrected line could rescue to the normal levels of neurons. So we could really prove that actually the patient-corrected line was able to rescue to the normal numbers of dopaminergic neurons. And interestingly, as we had single-cell data from these two, what we did was to just check which genes were up or down regulated. And again, the mitochondria had a role here because there were different genes implicated in the electron transfer that were down regulated in patients when compared to controls. So the mitochondria seems to be a bit everywhere because it explains this difference in patients, but it also explains some differences between the 3D and the 2D. So that's kind of the big main messages we could extract from our data set, and we wanted to include or integrate this data set into 6 other published midbrain data sets. So what we did was to integrate our cells with all the other studies, bringing together 190,000 cells using mutual neighbors implementation from Seurat. The results was that the cells were aligned upon their origin, so either 2D, fetal, in vivo, or post-mortem, except for one of the datasets, the Zagaria 12, which was a bit of an outlier. So it didn't actually align to where the 3D cells should be expected. After trying to build a kind of consensus annotation, which took me some time, because each dataset reported cells in a different way, I managed to put everything in a kind of similar grammar. And the main message is that the neuron abundance is much more stable in vitro than in vivo. This could be for many reasons, but it could be also an artifact. So we have to think here that whenever we try to sequence at single cell level neurons, they are much more fragile. So trying to capture bigger and more fragile cells might be very prone to error. So the fact that we have much less neurons is probably biological, but it's also technical. So if our study just wants to focus on neurons, we probably need other techniques like single nuclei if we want to be a bit more successful. But in our case, we did single cell because we were also interested in seeing the mitochondrial genes. So yeah, there is, I think, a compromise that's not very well discussed. whether to use which kind of approach should we use if we want to capture all the subtypes of neurons that we have in the dataset. So again, as we have previously seen, we wanted to see if we could capture some kind of developmental temporal axis in the integration. And just by coloring by differentiation day or post-conceptional week or gestational week, we could see that cells move from left to right. It's just by plotting this one. So somehow the in vitro samples, the first trimester fetal samples were preceded by in vitro samples, and the second trimester fetal samples were preceded by the first trimester. So clearly, there was a kind of developmental axis in the first UMAP component. To confirm that, to actually see if there was something interesting here, we ran 3 different cytotime tools, Destiny, Monocle, and Slingshot. We ranked the cells based on their cytotime values, and then we performed pairwise correlations to see how these tools agreed. Destiny and Monocle 3 agreed quite well, so we decided to use Monocle 3 for downstream analysis. And actually, that's the result. So here we can see UMAP, where cells are colored by cytotime value, and the blue colors indicate kind of lower cytotime values, where midbrain progenitors are. And the other colors, yellow and orange, are showing different lineages or branches that we can get, including astrocytes, but also neurons. But what was interesting about this was that once we got the cytotime values for all cells, we could also look at the cytotime values of the neurons themselves. And within each data set, we could see that the cytotime value actually increases. So the cytotime is not only capturing the differentiation axis, it's also within neurons, there is some kind of maturation process that's being captured by cytotime. So the samples that come from older ages are actually associated with higher values, which was something that we didn't expect, not necessarily, because there is a difference transcriptionally that is also captured in neurons. So once we have this developmental axis defined, another thing we would like to do is to understand these dynamics. For this, we identified 50 gene modules of co-regulated genes whose expression changes with cytotime. And to identify these underlying modules that we obtained, we computed activation scores to find whether they were linked to certain identities. And at least the half of those modules were very clearly enriched for certain identities. As in our case, we were interested in neurons, we focused on the top 5 highly activated modules in neurons. And those are the ones that are listed here, except for module 1, which, as you can probably see, it's mostly linked to ODC. But all the others are expressed in neurons, but also in other cell types, mostly mature neurons, and also I think it's some progenitors, which is quite curious. So the next question is, what do these modules mean? What are these modules doing? So we had to somehow annotate and give a functional biological role to those modules. And we performed Go enrichment of the genes that were contributing to each module and found that module 6 is linked mostly to synapse, module 12 is linked to calcium metabolism, and module 18 is linked to axon terminus. The other 2 modules, 21 and 46, could not be linked to any kind of biological function, but that doesn't mean they don't do anything important. We found something interesting for module 12. In this case, we could see that the older the samples are, it seems that they actually have a higher activation for this module, which might indicate somehow that there is a more important role of calcium metabolism during maturation, because this is done on neurons. So I don't know if that is in agreement with current literature, but it seems that somehow the calcium could be more important. Another question that we had in mind is whether there was a good or a bad alignment between the iPS-derived models and the fetal tissue. For this, we performed pairwise correlation of their expression signatures. And what we saw is that iPS-derived models start to diverge from fetal development around 10 post-conceptional weeks. This is somehow when the red in the heat map turns blue. And when we look at the maximum correlation, is essentially between gestational weeks 6 and 8 with organoids that are between 15 and 70 days old. So that was what we found. So there was a very good alignment transcriptionally between first trimester fetal samples and the organoids of around 60, 70 days. In contrast, postmortem samples had a very low correlation with all of the other samples, except for the outlier dataset I've mentioned before. So there is something weird about these 3D organoids, which are more close to the postmortem rather than to all the other cells that we have profiled. So although with single cell we have identified significant changes in the expression dynamics, now we need to incorporate the spatial context to better understand the trajectory of midbrain development. So that's the reason why we perform spatial transcriptomics, and we perform it on different sections of fetal samples from 7 to 17 post-conceptual weeks. And in the case of organoids, we also generated sections from 40 to 120 days. And those are the sections we analyzed. As a kind of quality, very brute quality control, we used different canonical markers to separate the different spots of the spatial transcriptomics data between postmitotic and progenitors. And as you can see in fetal samples, the postmitotic neurons tend to populate the intermediate and mantle zone, while in the organoids they are located at the external area. The BZM technology that we use in this project captures the RNA of tens of cells, not at single cell level. So to be able to know which kind of cell type we have in each spot, we have to perform cell type deconvolution. In the panel above, we show the distribution of one of the dopaminergic subtypes we were discussing before, the subtype 2. And in green and yellows, you can see the regions where there is more density of these neurons, which actually correlates very well with the Th positive neurons. So once we know the location of those neurons, we can actually try to infer spatiotemporal trajectory during development. At 7 post-conceptual weeks, as said, neurons are mostly in the ventral, in the more ventral area, while at 11 and 17, they are located around the ventral midline. So George, who was the one that led this analysis, decided to try to draw manually different potential maturation paths, and then superimpose and analyze the gene expression dynamic of those maturation paths and see if there were some gene dynamics that could be interesting. The results are somehow summarized in this panel below. Here we can see that in the case of TH, there is an increase in organoid expression, but there is a decrease in this maturation path in the fetal samples. There are other markers like SPOC1, which is important for, I think, the extracellular matrix integrity, which seems to be upregulated in all models. And then we found kind of similar trajectories, but always with some outliers for mitochondrial genes, which seem to be important for the maturation. Something else that the spatial transcriptomics can provide is investigate the cell-to-cell communication, particularly using this kind of ligand-receptor interaction between neighboring spots. Being able to identify this activation of ligand-receptor pairs allows us to give an idea of the pathways that are actually activated. This is an analysis done on 11 post-conceptual weeks, and in this case, we observe a lot of highly activation of communication pathways. But more interestingly, among the top 20, we found an enrichment and overrepresentation of ligand receptor pairs that were linked to the heparin growth factors, which is known to be involved in neural maturation. In this slide, I'm showing one of these examples of ligand receptor activation, which is the MDK ligand and the PTBR-Z1 receptor. And they were mostly expressed in around this area of the ventral midline. Not all areas have it expressed for either for both, but in this ventral midline, you can see the yellow and green colors, which indicates that they are both somehow expressed. And finally, we use the resolution of the spatial transcriptomics to quantify the spatial similarity between the fetuses and the organoids. And for this, we use to tell what was the similarity between those samples, we ask, who are your neighbors? So we essentially quantified from one given spot who are the identities of the neighboring spots. And by using this metric, we calculate the spatial similarity between all possible samples. What we found, and this is shown in panel E, is that the day 40 organoids was somehow correlating relatively well with the fetus samples of the 7 and 11 post-conceptional weeks, while the organoids of day 70 and day 120 were correlating more with the post-conceptional week 17. Apart from that, we could also estimate the fetal age of the organoids, and that's what's done in here. And by doing so, we see that the day 40 organoids has an age of 11 post-conceptional weeks, while day 70 and day 120 have around 15 post-conceptional weeks. We were a bit surprised about this result because it's showing essentially that whatever we do with the organoids between day 70 and day 120 is not actually improving the maturation. So we need to change something in the protocol if we want to go back at this, because that's not increasing the age of the organoid. So that's pretty much everything I wanted to say today. We have seen, I've shown you this kind of detailed molecular and cellular profiling of the first 2 trimesters of the midbrain. We have seen with histological images that the midbrain complexity emerges during the 2nd trimester mainly. We have seen that there is a biological recapitulation of 3 models versus fetal tissue, but that's restricted mostly, at least transcriptionally, with the 1st trimester. We have seen that the spatial transcriptomics is a tool that is able to show conserved differentially expressed genes along developmental pathways and actually bring up some relevant ligand receptor pairs that are actually indicating some activation of some pathways. And finally, in the wall of more disease modeling, we could link the DTDS progression with the neuronal loss and the mitochondrial dysfunction. So that's all. I want to thank again all the people involved in this project, the shared co-authors, the PIs, other authors that have tangentially contributed, as well the patients, which is the one that give, at the end of the day, purpose to our work. So yeah, that's all I wanted to say today. Thank you. If you have any questions.
Thanks, Pau. So I have one. If you could rewind to where you showed the later time point of where the 2 patient lines showed reduced numbers of neurons, not at the early time point, but at the late.
Yeah. This is here, I think, right?
Yeah.
This one?
So I guess I have a ton of questions about the different lines, about when and where maybe we can start there. When does the dopamine transporter turn on? Is it expressed in the progenitors? Does it wait until the neurons are made? Do you know that offhand?
I mean, I can tentatively answer that, but I think Ron knows better than me, so.
The
I don't know actually, but it's a good question. I mean, a related question is, is it dopamine that's killing the cell? Presumably it's dopamine, right? This phenotype, which is so striking in the patient lines. So these lines are null for the gene, right? Both alleles are deficient. Yeah. So what that means, I guess, is that dopamine is being released, but it's not being transported. So it's not being properly
cleaned up.
Yeah. So
it might become toxic for the neurons there somehow.
Right.
Yeah, I was also noticing that here where they're doing okay at day 40 is 40%. So they actually not only don't progress, but actively. I mean, there's other things going on in organoid. Maybe there's more astrocytes being produced or something later too, but it's a pretty big drop. That's from 40 down to looks like around 10%. So like you said, I would guess there's not only I would guess there's some death going on too.
Mm.
Well, there must be, Carla, must there not? So in the controls, it's still around 40.
Yeah.
So
they've only moved up a little bit, right, from here to here, if this is the same range. Agreed. Yeah.
right. Another thing that's interesting here is if we're talking about this, is that this mitochondrial involvement is very interesting, because actually when dopamine is transported by the transporter, it's co-transported with sodium. Now, this is slightly above my pay grade, and some of you on the call may know more about this than I. But as a consequence, the cell depolarizes. So it activates calcium channels. And of course, the mitochondrion is critically involved in buffering calcium. So when a cell fires calcium channels, the mitochondria is where the calcium goes. So here's where my off the cuff thinking sort of breaks down. But it seems like this is a very interesting model for the involvement of mislocalized dopamine and mitochondrial, I don't know what the right expression is. So what do you think is going wrong here? When you say the mitochondrial respirosome is downregulated, it seems like, does that mean that mitochondria are downregulated?
No, I would say that the elements that are part of the electron transfer chain, there is something that's less expressed than in controls. Which I think that here it's where we should look individually to all the genes that are annotated under those processes and see what's actually downregulated, because I'm pretty sure it's not all of them, but there might be some components of this chain that are more critical than others, and that could be interesting actually. Because enrichment is always, yeah.
And the mitochondria, just to make another very general comment, if we had Richard Ewell on the call, who's at NIH, he's done a whole lot of work, his group, in showing that mitochondrial dysfunction is a critical part of the pathology in Parkinson's. So this is an extremely interesting topic we're discussing here, and the fact that you appear to be able to correct it with the CRISPR manipulation suggests that this is actually maybe a very interesting model.
Yeah.
Actually,
I didn't think about the link with calcium, but obviously it's mitochondria. Calcium is very important. It goes there. So yeah.
Right.
Yeah, it'd be interesting for calcium
Carlo,
imaging for another reason. Go for it.
Don't take amphetamines. Really, it's not a good idea. Even though it might make you feel good temporarily. This whole system is disrupted by amphetamines. Yeah. Okay. Related question, maybe while you're go for it, Alex. Yeah.
No, please.
No, I was just going to change the subject a little bit to OTX.
Okay. Well, I guess piggybacking on the calcium questions, I think you went over it so fast, but you mentioned some kind of calcium gene network during development early on. And it seemed like you had some thoughts on that, and I wondered if you could elaborate on it. And also a beautiful piece of work putting together all the 2D, 3D, and primary tissue and different techniques in one place. Thank you for that.
Yeah, thank you. What we saw with that module, that's actually a module that's obtained from the same monocle treat tool. So that's a module that's detecting genes that are correlated throughout developmental time. So these genes that change coordinately with developmental time. And what we saw in that slide is that for within a data set, when we increase the developmental time point, this module is more activated. So it's the genes there are actually more expressed. How that links to this story of the DDTS syndrome, I don't know. but probably we will need to look for the activation in the patient samples and see if that's actually showing that this difference that we have seen. I think that would be a very easy, quick check if there is a difference there. And maybe going farther, we need to see if this module are some transition factors which are regulating them and check that too.
Okay, and then I guess a blue sky question, like how mature do you think you can make these dopaminergic neurons in vitro? And, there's still this big gap between like the Moscow paper has like 10 adult substantia nigra molecularly defined subtypes. And, we're lucky in these in vitro studies if we can say it aligns a little better to substantia nigra than to VTA. And so how far do you think we can take it?
That's a good question, but I think it has two answers. The first one is, how do you manage to capture so mature neurons without problems with single cell? And then the actual response to your question is, well, according to the spatial, we can capture things that look like 15 post-conceptual weeks. Maybe we can go a bit longer than that if we refine our organoids, we change the protocol or somehow. But the funny thing is that just the transcriptome says it's before. So what is telling us a better picture here, spatial or single cell? I don't know. So I would say around 15, but 15 to 20. But there is people that claims that you can actually go a bit longer. I don't know if that's possible, honestly.
But the other
Did
thing
you
you
look at
noticed
the-
is that the last 40 days of your protocol, or I can't remember how many, didn't get you
No.
further down the road, at least by the one global metric you
Yeah.
had.
Yeah.
Sorry, Alex, you have another point there? I jumped in.
Oh, I was just going to ask, I missed it, but did you look at the profiles of the xenotransplanted dopaminergic neurons from the Parmer lab?
No, I should have included that. Yeah.
We recently added those, and it helped us to map our organoid data maybe to kind of, daisy chaining on theirs to more kind of defined subtypes, because they have a few different time points and you can see what maps to substantia nigra versus VTA. But there it's still not, I guess the cell number is low, but it's still not getting to, like the SOC6 AGTR subtypes, et cetera, or the Geminin subtype that we're seeing in adults. Maybe if we left them for like a year inside of a rat or a mouse.
Mm-hmm. When looking at neocortical data, we saw a huge effect of xenotransplantation. But in neocortex, so looking at 6 layer identities, the transcriptional signatures of those different layers basically went from being able to see 1 or 2 deep layer identities in the organoids, despite the presence of the master transcription factors of the upper layers. The full transcriptomic signatures weren't coming on, but that really changed in the xenotransplantation. The environment makes a huge difference there.
Yeah.
What were you going to say, Rob?
Well, I was going to ask a question that's relevant to what Alex and you were just saying, which is when you see these differences, what do you think the signals are that are responsible for the maturation?
I'm afraid that to be able to provide kind of more clear answers, we need to increase the number of neurons to reply to this, because the number of neurons we got from this is not huge. So if you want to identify more subtypes and which subtypes are more critical and everything, I think we need to manage to provide more neurons, to sequence more neurons. And I think this is problematic, at least with 10x genomics. If you use single nuclei, it might be different. But
maybe it would be useful for people who are listening to say something quite general about why this is such an interesting topic. So I'm not sure if it's exactly true in humans, but as I was saying in this intro that I gave, when I asked Lars Olsen when you can first see dopamine, he answered, you can see vast amounts of dopamine as soon as TH is expressed. right? So why is that important? Because dopamine, and through its receptors, is a modulator of all kinds of subsequent cell biology events. So dopamine is very likely a critical regulator of all kinds of features of the maturation of other parts of the brain, right? So that's kind of what we're discussing here. So, and then there will be a feedback on the dopamine system, right? So I don't know if other people agree with me, but I think this topic is really, really cool, right?
I think in addition to xenotransplantation, something like a ventral pallium or ventral, ganglionic eminence assembloid with this kind of system might get you a step further.
Yes. Sergio Pasca, we need.
Right.
No, there's
get
no
them to
question.
talk to the cells that they usually talk to. To turn into the different things because they don't know what to turn into, right? They're still kind of floating in space. They know they're in the midbrain. They know they're dopaminergic, but they don't know.
Well, I think they know they're dopaminergic, but they don't quite know what flavor.
Right. Yep.
Right. Yeah.
Yeah.
Is that
I
right?
think that there are these like initial classes that are largely intrinsically coded based on morphogen delivery and position. But then there's post-mitotic state refinement that probably deals with these extrinsic signals. Gord Fischel has this nice CAPEX and Fischel review on this kind of nature, nurture, and circumstance model. And I think the best evidence for that in cortex was Larry Zapersky and Karthik Shekar with the visual deprivation showed that the post-mitotic state refinement of upper layer visual cortex neurons depended on activity. And that, there's 3 subtypes in rodent, and you would kind of see a generic subtype in between these 3 with visual deprivation. And, that may be the tip of the iceberg. And like you said, Ron, it could be that the dopamine inputs play a important role in many brain regions. And in that, and that's another thing we're missing with like region-specific organoids versus assembloids or xenotransplants. And I think the xenotransplantation is like the most extreme version of giving the extrinsic environment, and so it's worth testing there. The assembloids, it's just hard to get consistency across them and within them. Like we've made mesocortical assembloids, but the contact with the dopaminergic fibers is different across the organoids, and the size of the organoids is different in each one. And I don't, I guess we could profile with and without the dopaminergic input to see. But
did you have any indication that you got more maturation or more specific regional identities when you did the?
We never did like single cell in region specific versus region extrinsic comparison. We just found it a little daunting. We were also at the point of trying to compare species by regions, so it exploded in terms of the combinations we would need to query.
Actually, Carlo, that's a very good question, because one of another side project I am doing is to generate cortical organoids with vasculature and immunocompetent. And I wonder if we could do something similar in the midbrain, like at least vascular competent. But the problem is that whenever you try to generate those in the organoid, you might get contaminating identities, because sometimes you add VEGF or something that for certain of the lines push them into completely different identities. So it's difficult to be able to bring all of these systems together and not have problems, especially with donor heterogeneity, because there are some donors that react to this differently than others. I know that the Parmar that Alex was mentioning, the Parmar scaffolds that they used, it's a kind of showed improved, but I'm still thinking whether we can, before turning into assembloids, if we can actually improve the organoids. Yeah.
Well, to go back to your data. So you had a very interesting slide where you showed the timing of the sort of peak of expression of TH, and OTX2 appeared late.
Yeah.
And I thought that was worth a comment if all of us are still tuned into this, because the There you go.
Yeah.
I mean, because here, if you look, for example, at FOXA2, right? I mean, my view is, I think they're generally shared, is that FOXA2 is responsible for the initial specification of the ventral midbrain and for the expression of sonic hedgehog. And which you can also see over the right here is early. And but TH, which was here, down below, is it peaks in the middle of this time course, along with DDC actually, and ALDH1A, which is an enzyme that's synthesizing retinoic acid. So, which is very interesting. I mean, there's all kinds of things we could say, but it seemed to me that the one comment I wanted to make is that your late expression of OTX2, you commented on it, is indeed very interesting. And but it's not the only place that OTX2 comes up late.
Yeah.
OTX2 also comes up late in some thalamic neurons in the brain. And so I just wanted to sort of reinforce you there that that's actually a really very interesting finding.
Good to know, because at the beginning when I saw that, it's like, oh, this trajectory is not good. But yeah.
If you take an antibody against OTX2, do you see this in the midbrain? Do you see peak of OTX2 late? Or I don't know whether you'd say late, but at a later stage than engrailed or FOXA2?
I remember that whenever this appeared as a result, we discussed to do it or not. We never do it, but that's something that remains to be done because I think it's interesting to follow up this.
Because in terms of maturing the organoid, right, that you were just discussing, right, it seems to me this is a clue that you want to sort of pick up on.
Yeah.
Right?
Yeah, absolutely. Absolutely.
Anyway, thank you very much. It's an extremely interesting topic. I mean, I could go on. Yeah. Might be worth saying something quite general if we're still all interested in this. So one of the things that I find, especially in this setting, Carlo, that you have taken responsibility for continuing, is how almost in every case the discussion could go on longer. And it seems to me that it sort of poses a general question for us in the field, which is how do we really most efficiently move forward with so many components to the developing brain? I mean, I would argue, in response to what Alex just said about the difficulty of assembloids, it seems to me that the initial assembloid that you would want to test here is actually between midbrain and ventral forebrain, striatum, not cortex.
I would guess that's easier just because of the robustness of connections, just to get it to work. But yeah.
Oh, there's well, because we know there are feedback there, right? There are different dopamine different ventral midbrain projections back to the ventral forebrain projections back to the midbrain. So yeah, but the point I wanted to raise with you all is, and Erin, you're on the call, I see, I think we're asking ourselves almost like a logistical question here. How, as a community, do we think it's best to promote these kinds of progress in this field? So we're not just stuck with a kind of, a meme that we're all trying to pursue all the time. Yeah. Okay, I'll leave it at that.
I have a picture to share here. This is the Lamano data grabbing their ventral midbrain subset. And I believe this is this the right transporter 6A3?
Yes.
So it is not expressed widely at all. It looks like there's a tiny little pocket of neurons that's actually expressing this, which is kind of insane to think that presumably your organoid is similar, where most of the neurons are not even yet expressing this. But the ones that do and its function clearly are central in the survival of tens of percents of these neurons. So maybe the whole organoid fills up with dopamine because the small number of neurons that are actually moving dopamine back out of the synaptic space or the extracellular space aren't doing that.
Yes,
but it's
you're making a really cool point.
But I'm surprised to see this. I would have thought it was kind of like TH, that, you know, once you get to play with dopamine, you got to be able to move it back in. So turn on TH, make dopamine, and turn on this transporter, but evidently not. But
well,
I guess
not in every cell, Carlo.
Right.
A subset of cells can do it for you.
Yeah,
This is a complex computer of cell types interacting with each other.
Yeah, they don't all need to have the transporter moving dopamine back out. You know, I don't think people call it a neuromodulator anymore, but that's kind of why they were spoken about differently than glutamate and GABA, right? That they and tone, they use that word tone, right? Having dopamine around at certain concentrations changes normal excitatory and inhibitory signaling. And this is kind of getting at that, right? It's not that every synapse is lined with dopamine transporters on every synaptic thing to control the exact super fast transmission, but to control the tone across what we might consider tissue rather than cells. This would point that direction.
People anyway. Use words like this because they don't really understand the pharmacokinetics of neurotransmitter function. Right. So you may correct this, Pau, but isn't this transporter predominantly presynaptic? It's present on a number of different membranes.
That was my belief too.
But anyway, the point really here is not to answer these questions definitively, but to really look for ways in which we can really change the field. So the point you're making here is a very interesting one.
Yeah, absolutely, I agree. And the fact that these the mutations from the patients I show are as a result of compound heterozygosity, it means that maybe there is something about combinations of mutations that might be more deleterious than others.
Carlo, can you just put OTX2 into this data set?
Sure. Yeah. And I think we can also look at it by age. Now this is all over the place. And that's, as we maybe could have expected, more, or at least quite heavily down here in the progenitors, but then also up here in the
but
neurons.
You need to draw one more arrow because it's where 6A3 was.
Oh,
specifically. Yes.
And that whatever that little subset of neurons is.
Correct.
Yeah, and OTX2 was late, right? That was the surprise you had for us in the middle there, huh, Paul?
Well, it's coming up late in that system.
Yep. As is the transporter.
Carlo, is this all time points together, or is just the later, or what is?
I think it is, but there might be a bias given that annotation. So let's see. Okay. It looks like it's only 14 and 15 that. So it's higher earlier, but this is just one day of range here. And so this was based on their annotation. So maybe earlier than this time, they said midbrain, but not ventral midbrain, and maybe they had a different annotation later. We definitely have more of the more time points. I was looking here. So these are all the different chunks of La Mano we have. Some of them are separated by region, as we saw, and others are simply by age. So there's a lot of looking to be done here.
Yes.
But I found this one, at least in the short term, to be the most interesting. So here's so as we saw, it was a little bit higher in the one day earlier than the other, but not necessarily because of the progenitors. Also, these predominantly blue or early neurons are high OTX2. So it could be there's two OTX2s going on here, if not more, right?
Yeah.
And you look at SOX2.
Real-time science, guys.
Yeah, that's very cool. See, I mean, so
Yeah,
the
again,
SOX2 is
little
still
bit of-
expressed here.
Yep. Here's the
Yeah.
progenitors and the glia, I think, down here, right?
Maybe
Yeah.
I should make another very general comment so everybody sort of who's taking the trouble to stay online is getting rewarded. I mean, another set of data that's extremely relevant here is all the work on the timing of neuronal differentiation. Predominantly, these studies have been done in cortical systems. And I'm very influenced by the work from Lorenz Studer's lab here, where they've defined epigenetic regulators of the timing of neuronal maturation. And I'll make one other very general comment just to make sure that everybody understands we haven't just completely spun off into our own little bubble. If you ask in primates, what is it that correlates most strongly with the prolonged gestation period in man? The answer is brain development. It's not the size of the fetus. It's not all kinds of other things that you might consider to be important regulators of the gestational timing. It's brain development. So the implication is that in humans, as opposed to other primates, this topic that we're discussing here, neuronal maturation, is key to being a human. There's something very special about it. And so in Lorenz's group have shown in two important studies that there's an epigenetic regulation of this timing. If you suppress epigenetic regulators, you can accelerate the timing. And so this is a bit like in contrast to what you were finding with your organoids, that between day 70 and day whatever it was 110, maturation seems to be stopped. Perhaps there, if you go in and manipulate the system epigenetically, you could mature the cell. This takes us right back to the topic we were discussing with Alex.
Yeah.
So I hope at least we're sort of showing that these things that we're discussing are really central questions. They're not kind of peripheral questions. Now, that sort of brings us back to the question of how best to get us all forward here, right? It's because it's not just our problem; it's the problem of the field that these issues are really difficult to pin down.
And to sort of layer on what you were saying, Ron, that not only is it about brain development, but by and large, it's about slowing it down, right? To be a human, you have to slow these things down. And that's exactly what Studer's studies are showing. Basically, how do you tear down these elements that have been programmed to slow that whole process down so that we can get further in addition in a smaller amount of time? But it's almost like reversing evolution, right? The whole one of the biggest points of building a human brain is do it slow, so you can learn for 20 years.
But why do you do it slow? Presumably you do it slow because in building a human brain, you're building circuits epigenetically. Activity is going to be involved, as Alex was saying, and telling us to go and look at Larry Sapersky's work on visual cortex. But you do it slow because you're building special circuits in human. That's why you do it slow. You don't do it slow because you just want to sit around. Right.
Yeah.
Anyway.
Very cool discussion.
Yes. Thanks so much, Pau and Ron. This has been a lot of fun.
Yeah, thanks, Pau. I need to go and look at there are a lot of things that you showed very fast.
Yeah, and then there is a bit of more work on the spatial space about the dopaminergic, which I have not shown. So if you have any further question reading the paper, like just get in touch, because there might be other relevant questions that can be quickly answered.
I would make a comment to you about this integrating the spatial data. There's some very interesting work that's sort of coming online now about using mathematical tools to extract spatial information and correlate that with the sort of like gene expression or morphological data that we're more used to looking at. And if you were interested, you could prompt me, and I'll go and find those references. That's kind of really kind of high-end math. It's rather nice stuff.
Sounds pretty cool.
Yeah,
Yeah.
it is. Okay. This is
maybe that's science fiction, but I'm always tempted to think about if we could develop an AI foundation model to try to answer or provide hypotheses about what's going on, because we seem to be stalled about hypotheses. So that maybe it's a time to think.
No, that's exactly what I'm saying. That's what these guys are doing.
I think we need more data.
Yeah, that's. But
at this point, even if you had every little bit of midbrain and dopamine in vitro data, your foundation model would be still data hungry, I would guess, at this point.
But you're not using the data in your spatial analysis. You're not using all the data that are there. That's what this tool is doing.
I have to read about it.
Yeah, exactly. Yeah.
Yeah, if you shoot me the reference, Ron, I can shoot it out to the email.
Yeah, I'll have to find it. That'll take me a minute or two.
Great.
Okay.
Thanks so much.
Thank you,
Namaste.
Pau.
Bye-bye.
Enjoy your last hour or two of sunlight for the evening still. Nice.
Thank you so much. Bye.
Ciao.
Thank you. Bye-bye.